Why not?
Hi, Dion. If you, it's good to hear from you. Thanks for joining.
I'm glad to join today. Thank you.
You're welcome. Yeah. We're a minute early, but we'll get started anyway. Why not?
Sounds good.
So thank you, John, for joining us as our guest. Just a reminder for everyone, this is a completely casual meeting. So if you want, you can turn your cameras on, turn your microphone on. John's going to introduce himself in McDougall Scientific and the topic, and then we'll just dive into a conversation. We've got some questions we want to ask John, but Dion, if you've got questions for John, then by all means, let us know. The one caveat is if you have any questions that are of a private nature, like IP related, we always recommend ask us after, because we are recording this and it's gonna be published live later. So, but feel free if you've got stats questions about a protocol or anything like that. Now's the time to talk to the expert. So John, I'll turn it over to you, and we'll go from there.
Okay. Thank you, Christopher and Heather. And thanks for inviting me here. This is kind of exciting. Never done one of these. See how it goes. My name is John Amrhein, Vice President and Managing Director of McDougall Scientific. McDougall is a niche, what we call a niche CRO. We specialize in data-related services for clinical trials. That's data management, statistics, and then some related services around application development, technology, project management, and so forth. We were founded by Janet McDougall in 1984. Janet was organized out of large pharma, but they needed her help statistically, so she contracted back to them. Not an uncommon story, but word got out what she was doing, and the business kind of grew organically. Janet always liked being a worker bee, so McDougall remained as a niche CRO, but that allowed us to specialize in statistics, and including, I'm finding or learning that we offer some expertise that is not widely available. It's in clinical trial design, innovative designs, and working with stakeholders in clinical research to engage with regulatory authorities like the FDA, Health Canada, EMA, to strategize product development. And we collaborate often with regulatory affairs experts in engaging the regulatory authorities for our clients. I like to think of it as the regulatory affairs groups tell us the pathway, where we need to go, and statisticians find an efficient way to get there. Meaning shorter clinical trials, fewer, lower sample size required, things of that nature. Mm-hmm. So that's basically what McDougall is, our expertise. We are headquartered in Toronto, Ontario. And we were recently acquired by Alimentive, another CRO in Ontario. They have a holdings company. So we have grown from, a medium-sized 20 individuals. Now we have a much deeper bench that, it's around 400. It's quite a change for us, yeah. Myself, I've been a statistician, ever since the 1980s, but not always in this in the life sciences. I actually started out in forestry, and they were teaching us sampling the forest and estimating timber volume from that. I was a disbeliever. I didn't believe that you could do such things with such little data. So I had to learn more, and I got into stats that way. Master's in statistics from North Carolina State University. Worked with the U.S. Department of Agriculture, and then moved to SAS Institute, and then to McDougall. And I was hired at McDougall as a senior statistician, and moved sideways and upward, maybe back down, and then sideways again to the point of being vice president.
Fantastic. Yeah, I didn't realize that you would use stats in forestry, but I suppose that's That's true. We never would have thought.
Everybody uses SAS or used to.
Yeah. Yeah.
Yeah.
Awesome. So yeah, hi Prem. Hi Andrew Skura. It's good to see you.
Hi, Christopher.
You guys joined a little bit before or after we mentioned this, but feel free to unmute yourself. If you've got questions for John or anything you want to talk about, by all means, unmute yourself and just chime in. The reason we started this meeting, I don't know, we've done probably 10 now, right, Heather? It's got to be like 10. You're muted.
Unmute.
There you go.
Sorry, dogs in the background. Yeah, I think we're going on sort of 8, 9, 10 at this point in time. Yeah.
Yeah. Well, it's because we all used to meet at industry events and network at the cocktail receptions, and we've not been able to do that for, well, since March was our last trip. And so sometime around June, we started this just as a way to keep talking to people and having casual conversations. So it's been fantastic. We've got a pretty decent group of people that join every month, and the list keeps growing. So if you enjoy yourself today, we hope you come back for the next one. So I'm going to stop sharing my screen so we can get everybody on here. Let me get rid of this. Perfect. So John,
Yes.
what can you tell us what the #1 thing is that a statistician can do to maximize the impact on a clinical trial? Heather and I have been working on, like, we at TrialStat, we're a data management platform. So we're not a CRO, we're not data managers, we're not statisticians or anything like that. We're a technology company, but we do work with CROs, and we've seen really good ones like McDougall, and we've seen really bad ones. And the bad ones have some pretty disastrous studies. We're working on one that has, I think, on their 8th amendment, protocol amendment, in a 6 or 7 month study, which seems extraordinarily high to me. So I'm curious to know, from your perspective, what is that #1 thing that you guys can do to improve the study?
Well, the #1 thing is it's a pretty broad category, and it's what I mentioned earlier, is efficiency. Efficiency can come in a number of different ways. You can reduce sample size. You can collect the same amount of information. When a statistician has a definition for information, it's whatever you're estimating. In oncology, progression-free survival, it could be a pain score, whatever it is that you're studying. Efficiency is estimating that value and how confident you are in that estimate, which comes down to how much variability is around that estimate. And efficiency is reducing that variability to give you higher confidence in your estimate for fewer patients, or with the same amount of patients, same number of patients, you reduce that variability. So you can keep the variability and reduce sample size, or keep the sample size and reduce variability. We do this by talking to the stakeholders, the KOLs, the scientists, the product managers, who have a lot of information about what has to be studied, who it has to be studied on, under what circumstances, what's practical as far as collecting the data. We talk to all of these folks, and we learn ways that we can reduce that variability through design features. The design features, some of them I'm sure you're familiar with. They include things like stratifying randomization. It could be having run-in periods where you want to, for like in pain, you want to tease out or remove individuals who respond to a placebo. It might be measuring a biomarker that's predictive or prognostic for what the population you're studying. There's a lot of different tools in our toolbox that are available for us to provide value to the client, to the sponsor, if we are brought in at a time where we can still make make that difference, make those suggestions, and improve the protocol.
Mhm.
And that's the topic of this discussion, is talk to us earlier rather than later. I haven't always been in this area, as I mentioned, in life science and clinical research. I've been doing it now for 13 years, and that's given me enough experience to have seen many protocol come into McDougall. Some of them, and I can tell pretty quickly whether a statistician was involved or not, because we are not involved in all of the study designs that we service. We provide the other stat services, tables, listings,
Right, that's good.
figures, and CDISC data sets and so forth. But I can tell whether a statistician was involved in designing that study or not. And there's at least a handful of times where we look at a protocol and you think, I wish they would have come to us sooner. Sometimes it's too late. Sometimes it, if we raise our concerns, then it causes one of those protocol amendments before the study even gets started. But it's better to do it then rather than later. So I always think of Homer Simpson's advice to his son Bart. There's 3 magical phrases you need, Bart, to get through life. I don't remember all of them, but I remember one of them, and it was like that when I got here. Sometimes I receive a protocol and I think, and then down the line it's like, don't blame us. It was like that when I got here.
That's a good point. So, so when is, you mentioned time, when is the optimal time for you guys to get involved in an ideal scenario?
Well, we have one repeat client who's gotten used to working with us, and they call us up when they have, and this is a client that does many, many, many trials. They come speak to us when they have an idea. So it's in a concept
real early.
phase. Very early, and we flush it out with them we take that concept into a synopsis. They take it away, have it reviewed by other stakeholders, come back. It either proceeds or does not, often with some improvements based on that feedback, and then we take it to protocol and so forth. Most of the time, we receive a synopsis, and that's early enough, because then someone's done some work flushing it out.
Yeah.
The protocol is not, as long as the protocol is not final or not near final, then that's a good time to talk to us. It's still, if you're still open to suggestions to changing a study. And I can give you an example. It doesn't go back too far. It came to us, a client who was studying, I think it was ADHD, and so the target population was fairly young, and their clinical phase studies went down to 6 years of age. And the regulatory authority came back to them and said, we want you to study children 4 and 5.
Mhm.
Well, there's no need to pretend that you don't have a lot of information already. You've done phase 1s, multiple phase 2, 1 or 2 phase 3. You have tremendous amount of data. And a statistician using Bayesian methods can leverage that to make your study of the 4 and 5-year-olds very, very efficient. And pediatric studies is one area where that kind of efficiency using Bayesian methods is becoming quite common.
Interesting. So, yeah, I never thought of it before, but I guess, yeah, you can rely on those prior trials to actually inform an upcoming
Mhm.
trial.
Yes, and we had another situation where a client came to us, and the study was, this is another, it was like that when I got here case. The study was just about complete, so 5 years. And they were getting ready to go to the regulatory authority, and the regulatory authority says, well, we told you that we had concerns about your study design as far as establishing efficacy. that this is a single arm study. We have concerns. And we said, come talk to us. And this was at the protocol stage. And they never did that. So fast forward 5 years and the regulatory authority is saying, well, wait a minute, no, so your design is not adequate. It was a single arm study. And in oncology, you can't, if you're using progression-free survival, any survival endpoint other than overall survival, then a single arm study is not acceptable. So we developed a design to try to rescue this study and construct what we call a synthetic control arm, so that 100% of the control data would come from a source other than the clinical trial.
Can you elaborate on that? Because now that you've mentioned that, I have not been able to actually wrap my head around what that actually means.
Which part?
The synthetic control arm.
The historic. Okay. The synthetic is I'm not sure I like that word but the synthetic means what others might call external data. Anything that comes from a source other than the clinical trial itself. So this could be your own data from an earlier clinical trial. That would be called historic data or external data. You're planning a phase 3. You leverage data that was collected in phase 2 as part of phase 3. That's a use of historic data. It could be from medical records. So instead of, and this was one idea floated in this case that I mentioned, that, well, let's go into some medical records and from a chart review, collect data on what would have been control patients. So that's another source of data. You could also just do a literature review. And it has to be done in a very rigid, logical fashion. There's literature out there on how to conduct a systematic literature review so that it's unbiased and you don't cheat. But you scour the literature, and including what they call gray literature, which would grab some results of other clinical trials that maybe failed and weren't published. So you overcome that publication bias. You conduct a systematic literature review, conduct a meta-analysis, and that becomes your external data. And you could do that for a control arm. Now, these methods are often used to augment data that you collect in your clinical study, again, reducing the sample size required for you to recruit in your clinical study. So, okay, so let's do that. Let's collect some control patients, but let's not collect 100 patients. Let's cut it in half and get half from some external source. The FDA calls, they have a new acronym, well, a couple years old, CID, Complex Innovative Designs. And
that's what they call it?
A lot of, yeah, Complex Innovative Designs. March 2020, 2 weeks pre-pandemic. Janet McDougall and I were in suburban Washington, D.C., Rockville, Maryland, at an FDA workshop. And the whole workshop was on complex innovative designs. And and they are encouraging and accepting some pretty wild ideas.
Really?
Yes. They have a program that you can submit. Here's my complex innovative design. And if they like what you're doing or they're interested in it, they will become part of your team to design your study. Which is, I mean, you can't get much better than that, to have FDA right there.
Well, yeah, no kidding.
Study, I mean, if you're targeting FDA in the end, anyhow. So
then, now that you mentioned that, like I've always heard the FDA, you know, at every industry meeting they always say, you know, we're open we want to talk to you, talk to us frequently. You're better off to get us involved early than later. From your experience, is that
That sounds
true?
familiar. Yes. Yeah.
In
With FDA
reality,
and
that
Health
is
Canada.
true. Yeah.
Yes. Yeah. You have to. I've learned this. When you look at correspondence from a regulatory authority, you end up kind of trying to read between the lines.
Yep.
And interpret what they're saying. They'll never tell you what to do, but they'll hint. And when
Right.
they say we have concerns about your design, please call us. That's about as direct as they're going to get.
Right.
Well, we have run into that on a number of occasions lately where stats has been pulled in a little bit later and there's been a lot of rinse repeat. So this is really, really great advice because as Chris was mentioning, we're on one that's Amendment 10 and one that's on Amendment 8 for some projects that really did end up involving bringing stats in to talk about a lot of these revisions that were happening that people weren't thinking about those endpoints. So this is really great advice.
Yeah, and I didn't realize that the FDA was really pushing for those novel designs as hard as they are. Like that, I know.
Now, to
sponsors are sort of pushing.
To be honest, all their early examples and the presentations at this workshop were from big pharma. I don't know in our attendees here what kind of companies you work for, whether it's biotech or big pharma, but big pharma has the resources needed, and fair enough, they're at the leading edge of these innovative designs. But that doesn't stop the rest of us from stealing their designs and incorporating them into our own.
Right, right. Peter, nice to see you.
Hey, Chris, how's it going?
Very good. Michael, I see you joined. Nice to have you here too.
Thank you, good sir.
More familiar faces. I have a question, and this is because I am not a data manager nor a statistician. And I floated this question by you once, John, and you had an opinion. What is the difference between stats and data management? And Andrew Skura, feel free to chime in. Andrew and I used to work together. Andrew is a director of data management. So he can play the DM piece and fill in the blanks for me.
Do you want me to start off, John?
Sure, thank you.
So data management is exactly that. You're managing the data that comes in. You're trying to get it to a certain point where it's usable by the statisticians to run algorithms and what sort of all sorts of programming on it to ensure you have quality data. So the data manager is the one that basically pulls in that data from all different sources. We just heard about historical data and external data. That could be lab data, et cetera. And get it to a certain point where it's presentable to the statistician, where the statistician can now look at it and assign power. That's how I've always envisioned my job as a data manager, is to make sure there's a quality attached to it. Everybody knows, garbage in, garbage out. You want to make sure that you've got quality, good data that's usable, especially for companies that are going to expose that data externally to people that want to look at it or people that are interested in that company, et cetera. So as a data manager, I've always been the one to take a look at what I got, clean it, because data managers clean the data, and then have it presentable to a statistician, and then John rolls with it.
Right. Yeah, I like that, Andrew. To me, it's the key word is one that you said is quality. I've always called the site monitors who do SDV and the data managers, they are the gatekeepers of data quality. And if they don't sign off and check off that, yes, all the quality checks have been done, then statisticians have to sit and wait, right? And statisticians, they want quality data. They don't want to be starting in their programming and prep and analyses and discover discrepancies in the data that then you have to back up and fix them.
Yeah, that's why I found it always interesting to from this meeting to bring the statisticians in first and foremost from initial review of the synopsis
Mhm.
to ensure that they're on board as to what the endpoints will be,
Mhm.
what the endpoints they're going to be running all their programming on,
Mhm.
and target, and you know, if everybody's on the same page as to what is going to make or break this trial, especially from the initial inception of what the idea is, then I think having that statistician who essentially will produce the final product is absolutely key to be first and foremost in the mix.
Mhm. Yeah, when we started to adopt EDC, start the transition from paper-based data collection to electronic when I first joined McDougall, and we learned pretty quickly that a statistician should be part of the review of the ECRF requirements and part of the UAT and so forth. And in the reverse of that is this client that I mentioned earlier, where they bring us in on the concept stage very early, a statistician to participate in that. When we get to the protocol, we are now going to start having a data manager as part of the team to review the protocol before it goes to the client's approval committees. Because we learned that without that, we were having the protocol finalized, okay, to hand it over to data managers who write the requirements for the ECRF. And they come back with a list of questions and clarifications that are needed and say, well, why don't we do that before the protocol's finalized?
Right.
So yeah, they worked very much together. Now the statistician, so data management is quality of the data. The statisticians, very simply, we answer the research questions. So whatever that primary endpoint is, secondary endpoints, we are the ones who provide the answers.
Got it.
Basically.
I have a question for you, John.
Because, sure, Andrew.
So you did talk about historical data and using potentially patient charts, et cetera, to get the flow of the trial moving, I suppose. Have you been involved in taking a look at historical data from an analyte perspective, from say a blood panel or a lab panel, to ensure that you've got quality bookmarks for that particular analyte, whatever it may be?
The normal ranges.
Correct.
The right normal range. Yeah. We have not, but I thinking about your question, I understand the need to do that. Yeah, are those normal? I was stunned to learn that normal ranges change and they're not the same from one lab to the next. I still don't understand that. I can't wrap my head around it.
It's the bane of my existence.
Oh, I know, it's terrible. They're
Yeah.
they're all over the map sometimes. You've got
How can
from
that
different
be?
corporations, you've got different ranges for
Yes.
almost the same person standing side by side, you know.
Yeah. So you go to one lab and get a test done, and yeah, I'm all right. It's like, well, next time I'll go to this lab. And, oh, you're really in trouble.
Yeah.
I don't
And
get
it's,
that.
and it's tough when they have a site where they thought they were having one site with one lab, and then they get 3 or 4 more, and they're not sure how they're handling it. And they didn't plan for it ahead of time, which I think is really the point of a lot of what we're talking about, is what will you plan for ahead of time, and how do you build that into design to make it easier in the long term, and planning in terms of your study. But
Yeah, that would be an interesting analysis, though, Andrew, to figure that out. What should they be?
Right. Yeah.
That's where I'm going to become a millionaire right there.
There you go.
And Peter,
I've heard that the first million is the hardest, but yeah, I have no way of knowing.
Peter, and from your experience in terms of stats, do you guys get your statisticians involved early as well?
Ideally, well, usually yes. At the protocol development phase, they're critical in figuring out what the key parameters are, what the estimate of response would be, which then allows you to drive the sample size that you're going to need to show statistical significance in the study that you're looking at. So yeah, usually a statistician is involved at the very beginning, plus as well to try and figure out if the measure that you're looking at makes sense. Then you also need the clinician or the epidemiologist to look at it and say, well, is that a clinically meaningful difference? Because it could be statistical, but if it doesn't move the needle, or if the FDA doesn't think that it's will be enough of a clinical change, then it still may not matter, even if it meets the significance. So it's, yeah, they need to be upfront, and then ideally, if you can get all your tables and listings and your analysis set up at the beginning, it drives the timeline at the end a lot faster too, because then you can go through the analysis as soon as the data is clean, you've locked the database. So that's also pretty critical too.
Yeah, it's to me, it seems like common sense. But then,
one would think.
Yeah, but then not everyone knows common sense.
We have a write-in question that came to me through the chat, Chris.
Oh, great.
And it's so why don't sponsors get a statistician involved early if it's common sense? I'm not really sure, but I have come across folks in the life sciences and in my previous lives in other industries. Why would I hire a statistician if I can do it myself?
Yeah.
I've been told that directly. I said, well, go ahead if you can do it yourself.
But it's challenging to find a good statistician. Actually, we're actually looking for a couple statisticians, and it's not easy to find someone that has the right experience and can be independent. And are they programmers? Are they statisticians? Are they and ideally in the smaller organization, you want both. Someone who can do both. Do the stats and program as well.
Yeah.
In some of the bigger organizations, they may be more specialized, but if someone has that experience, then it's you can get a job pretty much anywhere these days.
I agree. Yeah. It's actually a good time to be in analytics.
Yeah.
And I think there's a fair number of I don't want to call them non-statisticians, or I don't want to be disrespectful, but a lot of people have had statistics training. They've had, most scientists have had statistics in university to a certain depth.
Right.
And I think some folks think that's sufficient to do the job of a statistician. So it's a case of, I don't know who said it originally. I think it's attributed to Donald Rumsfeld. But you don't know what you don't know. And this is some of the presentations I've given in the life sciences is here's some of the tools that we have in our toolbox, and not everybody is aware of them. And that's the value of a statistician.
Well, I liken it to programming, software programming. You can be a programmer. But programming is actually a form of art. And I would think that data management and statistics are very similar, because you can't just apply the practical components of being a statistician. You have to think and be creative, right? And so you may have the formal training and perhaps some formal experience, but if you don't have that creativity, then I don't necessarily think you're adding as much value to the process as you could be, right?
That reminds me of another quote, Chris. I don't know how many folks on here are Harry Potter fans, but if you remember Professor Snape, Alan Rickman, when he's teaching potions class, he said, I'm going to teach you the subtle science and exact art of making potions. And I jumped out of my seat in the theater and said, that's statistics. And they escorted me out of the cinema.
Yeah.
That's amazing. I love it.
I thought that was a very clever phrase, the subtle science and exact art.
Yeah, it makes sense to me.
And it applies to a lot of areas.
Most definitely. Andrew, were you going to say something?
Yeah, I think good data managers, because I know a lot of statisticians, but I never really got along with statistics at university. So it was never my forte, but I understood it. I know exactly what it was there for and why I was learning it. But it wasn't the path I was going to take. But as a data analyst, because at the heart of a data manager is a data analyst. They're always looking at trending. They're always looking at data as a whole and how it works and how it's put together, how amazing it is or how awful it is, because you can see that when you start looking at it and trending it in a certain way. I think there's a misnomer with looking at statisticians in sports. They tend to look at essentially trending of that data. They're not really statisticians. They're just trending it and creating patterns. And so the heart of a data manager is always someone that analyzes the data and is looking for that trending and looking for those patterns. And they tend to find them as they're inside it and looking at it. But the validation is when the statistician comes along from my perspective and says, that's what it is. That's what you're looking at. And there's your view. That's what you want. And so, I always, in my jobs, I've always wanted to be the person that gets it to such a point where I've trended as much as I can, and then it's just ready to be released.
Mm-hmm. So John, I read in one of your white papers about doing scenarios, and I didn't even know that was a thing. So can you talk a little bit about the various scenarios that you as a statistician can actually run for the trial at, like, when when you're designing the protocol? Because I didn't even know, like, it just never occurred to me that was a thing, right?
Yeah, that's part of simulation.
There you go.
So yeah, if under certain circumstances, and this is becoming more and more common, and it's becoming more and more common because of computing power. And there's packages out there that are supposed to be simulation packages that help you do this without a whole lot of programming. We just do it in SAS. But you might, it's usually done with the more complex designs. We simulated a design that's in one of those white papers where there were several unknowns. And you do this because you want to get some handle on what might happen in my study. So if I stratify it by a certain baseline characteristic, and I have an enrichment period, and a certain number are going to drop out for various reasons, and you put percentages around some of these. Okay, let's say if 30% are going to drop out, or 15%, and you specify all these parameters for unknowns. And then the statistician will put that specification, will put a distribution around it. And say, okay, so you might have 10% dropout, but that depends on what patients you recruit. It might be exactly 10%. It could be 6%, it could be 15%, 14%. So the statistician in the simulation program will add some random variability to all these different parameters that we need to specify. And it gets complicated if you have 4, 5, 6, 10 parameters, all with variability around them. There's uncountable numbers of combinations of the 100 patients you intend to recruit. But a computer, you can start it running, and it can run your trial 10,000 times. And then you just see what happens on average. And you say, okay, that's what we can expect to happen. That's what's most likely to happen in your study, given this scenario where dropout's 10%, and you're going to learn to lose a certain number through a placebo run-in, and this arm is going to do better than this arm. And then you change those parameters. Say, and that's another scenario. It's okay, let's say these two arms are equivalent now. And rerun it another 10,000 times. Now what's going to happen? And you can just play and run these different scenarios, and folks, and then you take it back to the subject matter experts, and they know what values of the parameters are really most likely. And you say, this is the scenario that pertains to what you told me is most likely, and here's what you can expect to happen with your study. There's a 67% chance that you're going to see the effect that you want to see. That your study design, your sample size, these parameters, given that there is an effect, there's a 67% chance that you will find it. And say, well, that's not good enough. What if I take it up to 125 patients? Okay, now that probability goes up higher. So it FDA does this all the time. Simulates things. And we've actually written simulation programs for interim analyses for what's going to happen the rest of the trial, and sent those programs off to the FDA. And then they play.
I didn't, yeah, no, that's a good point. I didn't realize you could do that. Again, not a statistician, but doing it at the interim analysis makes sense as well.
Yeah. Yeah, so we, we've done it. That one's quite common. It's called conditional power. Condition, you're halfway through the study. Given the data I have, how likely am I to get the result I still maintain that power, because my assumptions at the beginning may have been off. They were probably wrong, but now I'm halfway through, so I can tighten up my assumptions.
Right.
So do I need to increase my sample size now to maintain my power the rest of the study? Can I continue as planned with the same sample size? Should I quit because I have no chance? And we give probabilities. Peter, you've done this. You're shaking your head.
Yeah, pretty much
You're
all the
nodding,
studies now. It's always an
Yeah.
interim analysis. I would say 80% of the studies we do now. So it's rare that we don't actually.
Wow.
Yeah.
Okay.
So you do this routinely?
Yeah, all the
then?
time. Yeah, and
Yeah.
if you're not breaking the blind too, then you don't take too big of a power hit. So a couple extra subjects is certainly worth the risk or the extra cost for figuring out, like you said, John, if do you have any chance of making the endpoint, you can usually get a good handle. Or if you have, you're short 100 subjects or some or patients, then you can just power the study. So you're not throwing, you're not losing time and or have to run another study later. So it does help.
Right.
And John, you said you can do this.
Chris, I'll make one more comment.
Sure.
That's one of our tools that we can put into the protocol if you come to us early.
That's right.
Good.
If you try to insert it, if you come to us, the protocol is already approved, and we say, oh,
yeah,
well,
they don't like that.
You have all this uncertainty. You should be doing a futility analysis partway through using conditional power. Now, if you want that, that's one of your protocol amendments.
Right.
Yep.
Yet another reason to get you guys involved earlier.
Yeah.
And is there anything that SAS can't do?
SAS, it can't make me a good cup of coffee.
Because I know SAS is quite powerful, but I didn't realize you could do this kind of scenarios in.
Well, it's SAS is a programming language. So if you're an adept programmer, you can get it to do whatever you want. SAS has always been a build it by yourself philosophy. We'll give you the programming language tools to build it on your own. A lot of statistical packages that have come out since, or others, we build it for you, and you accept what we provide for you. So R is the same way. It's a programming language. If you're adept at using it, you can get it to do pretty much whatever you want.
So yeah, so that does bring up my next question, which is what, apart from SAS and R, are there any other software platforms, packages, tools that you guys use on a regular basis?
We use some other software packages for power analysis. There's a couple of them out there. We're pretty much a SAS shop. Some of our younger statisticians will use R for some experimental type of programming, but we require SAS be used for anything that's has to be submission ready or pass audit.
Right.
I guess I'm more the old school that if you're using open source software like R, you're going to have to validate it on your own. Whereas SAS is, they've already done the quality control on it. Well, when I worked at SAS, I learned that they use double programming.
Oh, really?
Before they release any procedures or any capabilities, they have their R&D person who develops the code, and then they have a programmer in their QA department who receives the specs. It's just like we do here.
Yeah.
And they have an independent programmer rebuild that code, and it has to match. It's the exact same process that we use. I doubt very much that R went through that. I don't know.
I wouldn't think so, no.
Yeah, we use SAS for most of these, certainly the involved stats, but I know we use SPSS for some of the more basic analysis as well. Not again for submission ready stuff, but we also do a lot of phase 4 research as well, and for publications and stuff like that.
Yeah, and there's others out there, Stata and Statistica. There's a number of good packages out there. I'm starting to see the word Python more and more, but I don't know what that does. Are there any Python programmers here?
It's a programming language for analyzing data. Just it's more like Power BI or business and like reporting and dashboards and that sort of thing. But from what I've understood, Python it's just a programming language that can do lots of different things with data.
Yeah, I mean, it's being a programmer, I'm familiar with Python. I never even considered people using it for this, but it is because it is a programming language. You can build anything in it. But it's like you said, John. It's only in the last year or so that I've heard people talking about Python for clinical trials.
Yeah.
Who knew? Have you ever received a protocol where you flat out refused to take the project on? I as a project manager have received project plans that someone else built, and I looked at them and I said, the cost can't be met. There's no way we can meet the timeline. Have you ever seen a protocol where you're like, this is just, we can't help it?
You're just gonna throw those grenades out like?
I guess none that I just walked away from, but I did a couple of them where I said this protocol has to be rewritten. And here's an estimate on us providing a statistician, medical writer, whatever, to do that.
So lots of caveats.
Yeah. I'm trying to think if we ended up working on any of those. I don't think we did. Right. And then I will tell you a story about one that's recent that it was more of a marketing diatribe than it was a protocol, study protocol. It had stuff in there about statements from Donald Trump and newspaper articles, and had all kinds of stuff in there. Why their product was the greatest thing to come along. That one was weird.
That was in the protocol?
It was in the protocol.
Was the My Pillow guy in there?
Well, they claimed that it had passed an IRB review, but I had my doubts.
That seems
So that
questionable.
one, yeah, that one, we're not moving ahead with that one.
Yeah, I guess I guess you can get all kinds of quality protocols.
Well, and
Without a doubt.
it's one of the exciting things about working with biotech is you get you meet and you correspond and talk to some very innovative, smart scientists and
Mhm.
clever thinkers producing these products, and it's quite exciting. And they come to you with different levels of knowledge about what it takes to get a product through the clinical phases of development.
Mhm.
Some are more receptive than others, but they're all exciting and different for different reasons.
Makes sense.
Yeah.
Another question. If you could put together a sort of a dream team of subject matter experts for writing the protocol, like by title, who would that be?
Regulatory affairs has to be there.
Mm-hmm.
The product, someone familiar with the product development, so a business person has to be there.
Mm-hmm.
They supply business constraints. Usually it comes in the form of that costs too much. There has to be medical people, the caregivers,
Mm-hmm.
the physicians, the surgeons, the doctors who know the patient population. They know what's practical. We've had some of them. I've been involved in protocol development discussions where they say, you can write that in that you're going to require a biopsy, but I won't be recruiting any patients.
Mm.
Because nobody's going to do that.
That's a good point. Yeah.
It's like, oh. But here on the other hand, we have the regulatory affairs or regulatory authority telling us that a biopsy is the only way that we're going to be satisfied with the measurement. Okay, now what do we do?
Mm-hmm.
There's a whole negotiation ensues. So that skill set has to be there. There has to be a scientist of some sort that is familiar with the mechanism of action.
Okay.
You need to know that. And ClinOps has to be there to know what's practical.
Mm-hmm.
That's my list, but I'd love to hear what other people have to say.
John, I would tend to agree with you. I mean, you want to have ideally, if you can, have all the different groups represented. Sometimes you don't have that luxury because they don't exist.
That's
in
right.
Smaller companies. So
that's right.
then you're kind of,
That's what I was thinking. Yeah, like a startup. Yeah.
yeah. So a lot of the smaller companies, there's a founder or two, and they have hopefully a bucket of money to spend on a study, and you've got to guide them the best you can.
That's true.
And then ideally, you want to talk to the agency as soon as you can, because they may have a completely different idea or understanding of what their expectation is. So you always need to come up with a good approach or study outline or design, and then they'll hopefully give you some constructive criticism, which they typically try to. And especially when you get into the more rare diseases and any orphan, then the agencies, especially the FDA, does is pretty helpful in giving you some advice as well, because they've seen a lot more products than any of us have, so it always helps to have that background.
That's an excellent point that I think gets lost, Peter, is that the regulatory authorities have access to information about other products in that therapeutic area indication that are being developed. And I think a lot of their comments and questions come from experience, information that we don't have access to.
No, for sure.
And I agree. That's an excellent reason to talk to them sooner rather than later, and they will provide the roles that are missing from your own team,
Yeah.
the physicians or medical folks, the scientists and so forth. Yeah, that's a good point.
And then as well, when you do have these meetings with the FDA, you really need to control the narrative and ask the right questions as well, because you can quickly go down a path that you're not wanting to go down. And they'll ask for a lot of things that you aren't necessarily willing or ready to put on your protocol or pay for. But unfortunately, one thing with the regulatory agencies is, they don't care what it costs and how impossible it is
That's right.
sometimes to, to find that one-eyed purple people eater, what do you want to call it, in your study.
Yeah.
And it's like, well, how are you supposed to find these people? They don't quite exist in real life. So it's always a bit of a challenge. So,
Yes.
or some of the clients really want to narrow down that patient population, which is great until you go to put your labeling together. And it's like, well, it's only about 2% of the population actually fit in that group, and it might work well, those 2%, but problem is you're not going to have any marketing success. So it's always a fine line how you design the appropriate, population that you're testing your drug in. So
Yeah, it didn't occur to me that the FDA would, and Health Canada would have access to information that everyone else doesn't.
Yep.
So you may not necessarily understand why they're suggesting what they're suggesting, but certainly coming from a position of higher knowledge, if you will. Yeah.
And sometimes it actually plays against you too, because as products are being developed and approved, sometimes it raises the bar for the next product, because you need to be better than what's out there, and they want to have something safer or some advantage, which obviously you need that for a marketing superiority or to be successful in the market. But they won't just let you get an approval just because you really. Then sometimes they ask for more and more data, and it gets prohibitively expensive to run these projects. So it is a challenge sometimes.
Yeah, we had a regulatory authority come back and tell us that the sponsor was choosing the wrong comparator. And they were shocked, but we think that's exactly why, was because they knew what was coming up very soon.
Oh, wow.
Oh, that's interesting.
Yes.
So a little nudge, a little wink.
Yeah, and so Peter's right. They can change things.
And was that Health Canada
more
or the
difficult.
FDA?
FDA.
Yep.
Yeah. Well, look at COVID. All the COVID studies that we're running, it's again standard of care, and the standard of care is actually a moving target, because as time goes on, new products are approved. It's added to the list, so it actually gets harder and harder for you to get your approval, because if the drugs that you're comparing against incrementally are getting better, then it actually gets more challenging for you to show that you're then as good as or superior to those other products. So it kind of raises the bar while you're actually doing the study.
Right.
So it's interesting.
Makes things even more complicated.
Very complicated. So yeah, not for the faint of heart, that's for sure. So
yeah, the COVID studies, just even from a practical perspective, have been very different from other types of other therapeutic areas that we've worked in. So with COVID, has the way in which these trials been run, has that posed a challenge from a statistics perspective? Because I mean, we're seeing patients being enrolled, for example, at any point in the day. Normally at a traditional research site, you're seeing patients enrolled, 9 to 5 kind of thing. But we're seeing patients that are obviously very ill, but they're being enrolled in the middle of the night at all times of day,
weekends.
So the challenge with this disease is there something about it that is causing the trials to be designed in a particular way that other therapeutic areas don't have to?
I would say it's more just the fact that you know, the hospitals, especially if you're in a severe or to moderate category of therapeutic treatments, you know, the sites are overwhelmed. And I think one of the bigger challenges is making sure there's that fine line between the sites being very busy and they have the patients to put on your study, and then being so completely overwhelmed, actually, they won't have the time to collect the data that you need. They'll miss some evaluations, and they put the wrong patients on the study. You know, there's one study we're working on, and there was one site, it was a super site, it enrolled like 35 patients in like a week. And these were severe COVID patients, but the challenge is a lot of the patients were dying because, you know, if you go look into the details, you know, they probably didn't have enough ventilators, so they didn't have any options for the patients. So they said, well, let's put them on this study because it gives them some kind of a hope that it might actually do something. But if they die in the first 4 or 6 hours or whatever, you know, the drug doesn't have time to actually take effect. So then, you know, you're potentially tying a death to a treatment when it's not really a fair comparison. And then RVs get concerned, and, you know, so it becomes very complicated to, you know, make sure that narrative is that, okay, it wasn't due to the drug. It was probably, and the problem is you don't see that until after the fact, because, you know, you haven't monitored the data and make sure that they met the real true criteria for the protocol. And the physicians can, you know, fudge the numbers or fudge the entry criteria a little bit just to say, okay, yeah, it meets that criteria, but, you know, did it really? You know, so it's, you know, it's a challenge. It's probably the biggest challenge with doing the COVID studies in the severe category. So
yeah, no, I believe it. They're moving. They're moving quickly. You know, they're moving at a pace that's fine, but these studies move so quickly that, you know, it's inevitable that they're going to change throughout the course of the study. Yeah. Well, we're on the hour. Before we wrap up, does anyone have any further questions for John or anyone else on the call, actually?
I have one quick question.
Yeah.
John, in your experience, what's the first thing you need to do when you find data that's obviously inaccurate, potentially is fraudulent, and you know it's going to create a whirlwind? What's the first thing you do? I know it's a tough question
It
because
is a tough question. It's
fraud.
fraudulent.
What do you do about
Fraudulent. So you're focusing that it's suspected fraud. Well, I think internally we would discuss it and get as many opinions as we can internally. So the statistical team, data managers, everybody gets together. And if we collectively decide, yeah, there's a concern here, then we take it to the sponsor and tell them what we found. So you're suggesting a case maybe where the site is fabricating data? Yeah.
We've seen that. Yeah.
Yeah, I mean, Peter sort of brought it up in the sense that, you know, you might have a super site where they're creating a lot of data, but it's potential that super site is overstating itself.
Yeah. Yeah, there's there are things to look for in the data that suggest when fraud is happening. It's commonly known that when people are making up data, fabricating data, they're not very creative.
No.
And for a site, the variability tends to be much lower in data values than you would find in other sites. There's also a measure which I'm curious about. I need to look into it and read about it more. Peter, maybe you know about this. It's in Junk Clinical, and they say look at the last digit because that's where there's a lack of variability. in the last digit of a number that's fabricated. They tend to end in the same number.
Really?
Interesting.
I didn't know that.
Didn't know that either. But it's
Wow.
certainly what I would
So-
suggest, if you have these super sites, you need to send your people in and the monitors in there as soon as they can to really get into that data, make sure that it is
Yeah.
appropriate and acceptable. And you can
Yeah.
bet for sure that if there is a site like that, the FDA is going to send their auditors to come knocking and is going to actually look at the data under a microscope. So, it is imperative, because if that data is missing, you may not have enough power, or it could, skew the data either one way or the other, which may not be favorable to your product either.
Mhm.
And it's very important. So you need to get on top of that. So, and then make a decision on whether you put a clinical hold on any further enrollment at that site while you investigate. And if they are inundated and can't keep on top of the data, sometimes you have to make that tough call, even though you want the patients. But it doesn't help if the site can't do what they're supposed to. Otherwise, it does affect the quality of the study, and that's not acceptable either. So.
Mhm.
Yeah, well, and I would assume you don't want to take the risk that it affects the quality of the rest of the data, such
Correct.
that they won't accept it, right?
Correct.
Even though
And-
it's an issue, you want to kind of deal with it quickly and efficiently.
Yeah, and usually you would put a sort of an upper limit on the number of patients any one site could generate on the study, because again, you could then have a big group effect and have some challenges with the data that you don't want.
Mhm.
And if again there was a problem at that one site, it actually could sink your whole study. So it's a big issue. So
Good question. Thanks, Andrew.
Yeah, that's a good.
Well, thank you, everyone. Thank you so much, John, for joining us, and everyone else who
discussion,
came.
Chris.
I really appreciate it. We're going to be publishing the next one for May over the next few days. So hopefully we'll see you all there for the next one.
looking forward to it. Thanks,
Awesome.
everyone.
All right. Bye, everyone. Have a
Yeah,
great
thanks,
night.
everybody.
Have a nice
Bye.
evening.
Bye.
Okay.
Bye.
Good night.